Let's talk about dihybrid crosses. Here are our learning goals. We're going to construct a Punnett square to determine the genotype and phenotype of the offspring from a dihybrid cross. We're going to predict the inheritance of simple traits of a dihybrid cross based on the laws of probability. So let's first talk about what a dihybrid is. Let's imagine we have a pea plant and it has two traits that we're interested in. One is height and the other is flower color. Here are the alleles for height: capital T for tall, little t for short, dominant, recessive. For flower color, capital P is purple and the little p is for white flowers, dominant and recessive. Alright, so we learned last time that a hybrid means that you're heterozygous for one trait. So if we were just talking about height, a hybrid would have one of each of the alleles for height. There would be a capital T and a little t. That's a hybrid, a heterozygous. Right? And the phenotype would be a tall plant. A dihybrid is heterozygous for two traits. The prefix di, means two. Hybrid, we learned, means heterozygous. So we are heterozygous for two traits. So what would that look like? For this particular plant, it would look like this. We have two alleles for height and two alleles for flower color. We just put them together. So we have a dominant and recessive for height and a dominant and recessive for flower color. So what would the phenotype be for this plant? It would be a tall plant with purple flowers. Alright, so let's take another case. In pea plants, peas can be smooth or they can be wrinkled. The allele for smooth peas is dominant, and we use a capital S. In wrinkled peas, that trait is recessive, so we have a little s. Another trait in peas is pea color. Yellow peas are dominant, so the capital Y is our dominant allele. Little y is our recessive allele, that's for green peas. Okay, so let's look and see how we would cross two plants that were hybrid, that were heterozygous for both these traits. Two plants that are dihybrids. Alright, this is what their genotype would look like. They're hybrid for whether they're smooth or wrinkled, and they're hybrid for whether they're yellow or green. Now we just show one of the parents here, but both parents would have exactly the same genotype. Okay, so the first thing we need to do is figure out what kind of gametes these individuals can make. Now, when we did simple hybrids, it was easy. Each trait produced two possible gametes. Now, since we have two traits, we're going to end up with four possible gametes. So how are we going to figure out what alleles will be in each gamete? Well, there's a simple way to do that. First thing we do is we take the first allele and we match it up with the first allele for the next trait. And that would give us a gamete with these two alleles. Then we match up the first allele with the second allele of the second trait. That would give us this gamete. Now we're going to match up this allele with the first allele for the next trait, and then we're going to match up this allele with the second allele for the second trait. And these will be the four possible gametes that could be made. This is every possible combination of these alleles. So our next job is to set up our Punnett square. Because we have 4 alleles from each parent, these might be the possible sperm, and these might be the possible eggs. We have 4 possible gametes from each parent. Now we need 16 boxes. Right? We had 4 before, now we have 16. Well, we're going to do this the same way we did the smaller Punnett square. We're going to take this gamete and we're going to put it in the box. Then we're going to take this gamete and bring it over and put it in the box. We're going to do that for all 16 boxes. Now we're not going to leave these alleles like this. We're going to match them up. We're going to put the alleles for each trait next to each other like this. And we're also going to put them in order so that we have the dominant trait, and then if we have a recessive trait, that would go second. So we go through and we fix our Punnett square and we fill in all 16 boxes, and this is what it would look like. The next thing we have to do is figure out what the phenotypes would be for all these possible zygotes. Well, let's look at it. Let's put a picture of a smooth yellow pea in each box where we have at least one dominant allele for each trait. We'll end up with 9 yellow smooth peas. We'll end up with 3 smooth green peas because we'll have 3 boxes where we have at least one dominant allele for the first trait and 2 recessive alleles for the second trait. We'll end up with 3 wrinkled yellow peas because we will have 3 boxes where we have 2 recessive alleles for the first trait and at least one dominant allele for the second trait, and we'll end up with 1 wrinkled green pea because we'll have 1 box where we have all 4 recessive alleles. Whenever you do a dihybrid cross, you always end up with this ratio of phenotypes. You get 9 that are dominant for both traits, 3 that are dominant for the first trait and recessive for the second trait, 3 that are recessive for the first trait and dominant for the second trait, and 1 that's recessive for both traits.